Today is Monday the 21st, and we're looking at male reproductive system, chapter 21. So, pretty simple system overall. The cells look a lot like cells everywhere. We're dealing with a lot of epithelial cells. Why epithelial cells? Again, why do we end up focusing a lot on epithelial cells? Well, because we're making something in a duct, and the duct is going to be secreting some kind of its fluids and reproductive cells, and so epithelial cell type is crucial for that kind of function. And so, of course, epithelial cells are going to be major players here. The other thing is that there are support cells. So just like we had talked about neurons and the fact that there are 10 times as many glial cells as there are neurons, there are support cells in the testes as well, and those are called Sertoli cells. And so without the Sertoli cells, you would not be able to complete spermatogenesis and spermiogenesis. So those are two processes that are involved with making sperm. Okay, let's see. So you can turn on the overhead while that's coming up. So we'll look at some of the structures that are involved. So there's the physiology part of it. What are the different components of the male reproductive system, and but this is a histology class, so we're mostly interested in the morphology of the cells and the cell types that are involved and how their structure is able to accomplish the functions that are required. I think there's a couple of things that are kind of interesting before we look at the first pictures. One is that the testes are sort of hanging out, right, in a scrotal sac. And the reason for that is because the temperature is around 34 degrees centigrade as opposed to body temperature, which is 37 degrees. And in fact, male reproduction won't work correctly at 37 degrees. It's kind of interesting. So some people speculate, well, the reproductive system is a very ancient system, and maybe the beginnings of reproduction were in cold-blooded animals. And so as we developed into, as organisms became complicated, warm-blooded animals developed later, they couldn't replace all of those steps, and so it was easier to just try to adjust the temperature and get it down to 34 degrees. One of the things that's typical too is that on the structure of the testes, you see, of course, a blood supply. So there's got to be arteries and veins and nerve. So we're used to seeing that now. It's as we've looked through organs in the body, that's pretty typical. You have to have blood going in, blood going out, and you have to have nerves, and they tend to be bundled together or in somewhat proximal location to each other, and that's what happens with the testes as well. Okay, so I think this looks like it's ready. Okay, so just some vocabulary here. Testes, this is where the sperm are coming from. The sperm are accumulating in the epididymis, which is this gland that sits above the testes. There's got to be a tube, of course, where this material can flow out, and so that's the ductus deferens here. So that's going to take the material that's accumulated in the epididymis, and the ductus deferens goes around here. You're going to add some seminal fluid, so you need a seminal vesicle, and there's an ejaculatory duct. And then this material is going to be squeezed out through here. There's a prostate gland here. What is this other tube here? It's not even labeled on the diagram. Yes, the urethra, right? So this is where you get, males are unique in this regard, is that we have shared DNA. So we have dual purpose for the urethra down here. So we have the bladder, so there's going to be urine here, but then you've got this this Y-shaped connector for seminal vesicle, and so that's going to be able to be eliminated, both kinds of material, through the exterior organ, which is the penis. Okay, as far as the, let's see, so we kind of covered everything there. Okay, let's see, there is a few things. This is the pubic symphysis. It's just this area here. Anything else? There's a urogenital diaphragm. Because of this Y-shaped connector, you're going to be able to control urine versus semen. Okay, so this is the testes in more detail. This picture up here is kind of annoying. It's kind of, but it's interesting how we were just talking about for the lab, whether it's okay to look at human sperm in a microscope in a college class, and I think so. I think it's kind of an important idea. Not to degrade the value of the sample, but actually to celebrate its importance and the we're interested in form and function, and so these are very efficient cells at what they do, and they're wonderfully designed, and to not look at them is a mistake. But anyway, so looking at the testes, what's going on here? Well, you've got a lot of tubules. The sperm is going to be produced in this kind of tubule. Why is that? Why do you need 25 meters of thin microscopic tubule? 25 meters, that's kind of a lot spread out. So why, we've seen this kind of theme before, right? Remember with the epithelial surfaces are often undulated, and small intestine is wrapping around. So in the case of the small intestine, we needed that surface area, right? And so surface area is, it's an important component again here, because you're basically dealing with epithelia, right? When you've got a duct and you've got some sort of lining cell that's epithelial derived or epithelial-like cell, that to have a longer, more surface area is a good thing. And so coiling up a bunch of small tubule is kind of important. And so that's what is going on in the testes. And then there's the arrangement of all of that tubule. So they're showing this in the diagram here. There's the straight tubule, where they finally labeled it a tubule, but the straight tubule is just a small collecting area from the seminiferous tubule. So the seminiferous tubule is all this stuff, and we were saying that all the seminiferous tubule plus the straight tubule That's 25 meters in length worth of stuff coiled up and packed in there. You know, the testes look solid because there's some layers over all of these tubules. So there's some protective sheathing here. What are those? Well, there's the parietal layer of tunica vaginalis, and there's a visceral layer. So there's the outer layer, the parietal, and the inner layer visceral of the tunica vaginalis, and that's protecting it. There's also the tunica albuginea. So there's these different layers that are protecting this arrangement of tubules. And then there's also they're separated into individual compartments, which is kind of interesting, but typical biology. It's kind of more efficient. If there's a defect in one, I guess maybe this is what, part of what was going on is if there's a defect in one, if you've got them segregated out, maybe that's part of it. If there was an infection in one or some sort of defect. Anyway, so they are separated out, and they're separated out by the septum here. So I think the total number of compartments was, let's see, the organ is divided into 250 pyramidal compartments. Reason why they're pyramid-like is because if you're wider at the around the outside and then they're facing in, they're going to tend to be more narrow in the inside. So that's why they use that description, pyramidal. We've seen that before. Okay, so then a whole one of these is considered the lobule. So then you collect the sperm that are being made. They're going through the straight tubule, and there's this part of the testes that's called the mediastinum testis, or the housing reedi testis. Two names for the same thing here. It's just a way of collecting this material and saying, okay, now we've got a localized area, thicker tubule than the seminiferous tubule, but you're going to need to store this material even in a larger extent. So you have these efferent ductules here that are collecting this material then and transferring it over to the epididymis. And the epididymis is, I think this is a structure that people don't really think of very often. We're all familiar with the idea that there's a testis and that there's sperm being made there, but it's going to be collected, and it needs to be collected into some sort of holding organ. And so this epididymis is holding some material, and then this material can be released into the ductus deferens and travel up, and we'll look at the rest of that in a minute. This picture here, so this is the histological picture. And there's a couple of pictures just like that, actually. Okay, what you see is kind of a circular arrangement. Why is it circular? Well, because it's a tubule. Right? So we're looking at kind of a cross-section of that. And you've got spermatogonia, which are the SG. Here, those are larger cells near the periphery. We have the Sertoli cell. And the Sertoli cell, as I was saying, these guys here, they look pretty similar, but the nuclei are a little bit smaller on the Sertoli cells, but they're a big cell. But they have this important supportive role for spermatocytes. So the spermatocytes, let's see, that's these guys. Those are spermatocytes. And we'll see what the difference is between a spermatocyte and the spermagonia up here. And then you have um, late spermatids, LS, and interstitial cells here. Looks a lot like like many sorts of tissues in the body, lots of cells, right? But there's some common themes here. The stuff in the middle is a little bit clearer in the middle, so that's typically a lumen, right? Any kind of clearing. The late spermatids. So spermatids are genetically set up to be sperm. They're not quite fully developed yet. Difference between a spermatid and sperm, and we'll cover that in a minute. But you see here that these cells are sperm-like, and so they're on the inside because this is a lumen, right? Because there's this is a duct, and so you're making something, and it's going to be it's going to make its way down the duct. How does it do that? Well, you've got these myoid cells here. Around this whole tubule, there's this thin layer. I don't know if you can see that, but I think you can. Kind of a one-cell layer looking type thing. And it's related to smooth muscle cell, but it's not exactly smooth muscle, but it has a contractile function. So it's pretty much functionally working like smooth muscle. So myoid, having this be coated with myoid gives it a contractile ability. Why do you need that? Well, you don't need a lot of force. Right? It's kind of interesting. We were saying skeletal muscle is designed for power, right? But a lot of smooth muscle function, you need some sort of motion, but you don't need such energetic motion. Well, you don't really need much force here, but you kind of want to encourage the sperm to make their way out of the tubules, and so having this myoid layer around the outside gives you some contractile ability. So that's one thing. Then the other thing is that these interstitial cells, or Leyden cells, they're surrounding the tubule. And their function, they have a very important function. They make testosterone. It's a big deal. The whole male reproductive system is kind of based on testosterone. Testosterone has been malaligned, simply. Maybe the source of all sorts of violent behavior, I don't know. But anyway, males need testosterone in order to function in a reproductive sense. So, so what cells are going to be making the testosterone? Well, these interstitial cells that are surrounding the tubules here, they're making the testosterone. How about in the development of the male sexual organs? Is that that's going to be dependent upon testosterone too. How does that appear? Well, it turns out that I think there's some cells from the mother that are, from the in utero, gonadotropin from the placenta is stimulating these interstitial cells to synthesize testosterone, and then that's going to lead to the formation of the ducts and the glands that make up the male reproductive system. So that's kind of interesting. Testosterone was needed to first form the whole ducts and glands to begin with. And then, in order to function as an adult, at the onset of puberty, you want the male to be able to produce sperm, and so then these interstitial cells become the factories for sperm. But then, where does the sperm come from? Well, where does the signal come from? There's a couple of steps in the signaling. Let's see. Here we go. signal for the interest. We're trying to. I'm just trying to think of how to present this on tracking the testosterone. So we were saying that during development, that the your mother is going to be in utero. That the gonadotropin from the placenta, from your mother's placenta, is going to stimulate these cells, these interstitial cells, to produce the testosterone. But for the adult, once it reaches puberty, there's it's triggered by luteinizing hormone LH, or also called ISCH for interstitial cell stimulating hormone. But usually, luteinizing hormone is kind of the one that people recognize. So. And then how is that triggered? Well, this gets into the pituitary thing. So what triggers the pituitary? Remember, the pituitary is the master endocrine gland, right? But then what's triggering the pituitary? If the pituitary is going to decide if it's going to start to enact a series of events resulting in activity of the male reproductive system to produce sperm, this master endocrine gland, the pituitary, has to be stimulated by something. And so that's the brain, right? And it's more specifically the hypothalamus. So the hypothalamus produces gonadotropin. And then you have the gonadotropin releasing hormone, and then you get the luteinizing hormone being released by the luteinizing hormone, and then you're going to activate these interstitial cells. I know I sort of mumbled through that, but you get the picture, right? So, I mean, it's a series of events, and these cells are really critical to maintaining that whole thing. So, in this picture, we're going to look at some other pictures that are just like this, but we're going to talk about the spermatogonia and how they become these late. You also have the spermatocytes and the late spermatids, but you don't see sort of all of the steps in there. And one of the reasons why you don't see all the steps is some of the steps are quick. So there's a first meiosis and a second meiosis. You don't see cells that are in second meiosis because they move so rapidly through the second meiosis that you only see spermatids. And the spermatids are these small nuclei like this. They're well on their way to becoming sperm, and they're more located near the lumen because they're going to be released. They're going to be released because the myoid has some contractile ability, and it's sort of squeezing them gently, encouraging them to go on their way. Once they get on their way, they're going down the seminiferous tubule. They're making their way to the straight tubule. They're collected into larger tubules, which are the mediastinum testis, or also known as the reedy testis, and then there is the efferent ductule. That there are multiple efferent ductules that lead into the epididymis, and that organ is a holding station for sperm, and then it's when it needs to be released, it's going to travel up through the ductus deferens and eventually make its way to the outside world. Okay. So this is another tissue section. This makes the point that the organ is divided into with these septa here. So we've got the seminiferous tubules, so we're talking about, which is 25 meters worth of these tubules, which we were just looking at the cross-section a moment ago of the tubule. These are all the tubules, but they're separated by these septa here, giving you the separate compartments, and they're pyramidal shaped because they're facing towards the inside, so they're narrower towards the center and wider on the outside. So M is the mediastinum. And here on this next slide, this is more histological pictures. We've got this one, and these two. And I think it's good to get, you know, the full effect of what these pictures are showing us. So the Sertoli cells, I see these are fairly large cells here, like that. You see that there's a lot of them, and they almost look like the epithelia. There's many more Sertoli cells than there's going to be spermagonia. The myoid here, that was that layer of smooth muscle-like cells on the outside, and then this stuff here was all the interstitial cells. So you see how, you know, this section looks quite a bit different than the other one, but I think it's good to look at that just because you see that things can look different just based on how you slice it, right? Because we started out with this picture, and we were comfortable with the idea that, yeah, this is a tubule cross-section, these are the interstitial cells on the outside, this is the myoid, smooth muscle cell-like. So, you know, that kind of picture versus if you look at this, then you're looking at higher magnification, and you're only looking at part of the tubule, so there's a lumen here, but you know, it's a different view of the same thing. But here you get much more of a feeling that this is kind of an epithelial layer, and you see how dominant these Sertoli cells are. Sertoli cells are an issue for male fertility too. So you know, a lot of the fertility doctors are interested in maintaining these cells. There's a guy who's, let's see, a chemist that I saw recently that I knew from a company that now works at the Buck. And so it was interesting to see that he is now kind of in our microenvironment here. And his company was supported entirely by male fertility doctors that were interested in culturing Sertoli cells. So if you can grow up Sertoli cells efficiently in a lab, there's a product there you can earn a living just growing Sertoli cells. Because apparently, I mean, since these cells are providing the support function for the actual germinal cells, the spermatogonia, that the health of the Sertoli cells determine to some extent male fertility. And so for certain patients that are having a problem, if you could add some supportive cells, or if you could culture those cells and then add some of these gametes to the culture, then you could maybe produce some sperm that could be used to have a child. So I don't know. Anyway, so the Sertoli cells are important. Just want to see you to look at this other view. Okay, so then what do you have here? Well, these are the interstitial cells. Another view. They're stained a little bit differently. And then here's the myoid here. Why all the little spots? Well, these are lipid. Why are there tiny lipids inside of these cells? Well, because what's the job of the interstitial cell? It's making testosterone, right? So it's making a steroid hormone. And so cells that make steroid hormones, they tend to have lipids because they're kind of lipid soluble. And so steroid hormone is going to be distributed in these lipid droplets inside the cell. And so if you see this kind of morphology, then that is a good guess on what it is. So keep that picture in mind. Lots of tiny lipids, and that's what you would need to produce a steroid hormone. And what steroid hormone have we looked at in some detail, and that would be testosterone. Okay, so then there's these two pictures, this picture and this picture, and these are produced by immunofluorescence. So you guys know what that is? Immunofluorescence, we talked about that in some detail. So it's usually indirect immunofluorescence, or IHC, or immunohistochemistry, IHC. So you're using an antibody reagent, and you have that reagent against a particular protein, and then you use a secondary antibody that has a fluorophore, and the fluorophore in this case is glowing green. So wherever that protein is, it's going to light up. So what protein did they use in this picture? They used, let's see, sulfated glycoprotein 1, also known as prosapocin. It's kind of interesting. The antibody reagents are so specific that you can get them to recognize only modified proteins, not just the protein, but a modified form of the protein. So the sulfated form of glycoprotein 1 is the prosapocin, and there's sapocin, and then prosapocin is kind of a precursor. It's sulfated, and the antibody recognizes only the sulfated form. Antibodies are now derived that only recognize phosphoproteins or dephosphorylated proteins also. So you can get them designed to recognize specific forms of a protein. So then you know whether the active form or a precursor form or whichever form you're interested in is actually being expressed. And so that's what's shown here in the IHC picture. This is for the Sertoli cell. So the sulfated glycoprotein 1 is on the surface of Sertoli cells, and these are in seminiferous tubules, and they're lighting up, and you see the extent of this. So just like the other picture that we looked at, we were saying, gee, there's a lot of Sertoli cells here. This guy. Yeah. So, I thought there were sperm being produced, and you're showing us support cells, and there's a lot of support cells. To show you how many support cells there really are, this IHC shows you that all the things that are lighting up are associated with the Sertoli cell. And then the next picture is a cross-section of just one seminiferous tubule, and then you see, what the distribution is, just in one, and you see that it pretty much takes up the whole area. Why is that? Well, because the spermatogonia, they don't exist without Sertoli cells. All of the genesis or growing up of these sperm has to take place with the assistance of Sertoli cells, and so they're well distributed all the way around the seminiferous tubule. So, the important process here is spermatogenesis. So, on a cellular level, there's the male reproductive system. We still have to talk about, the external male reproductive organ and all that. But on a cellular level, the testes is really the thing that that is creating the sperm. And so we're interested in sperm creation because that's what males contribute to the equation, right? There's the females are contributing the egg, the males are contributing the sperm. How do the sperm arise? Well, there's this precursor cell of spermatogonia. And when I say the word precursor, you should think, so is that like other precursors that I've heard of? Yes. And so, overall in biology, anywhere where there's a high demand on cells, there are stem cell progenitors, right? So, and a lot of it has been made of this lately about the stem cells and researchers are identifying stem cells, and we've taken induced pluripotent cells and embryonic stem cells, and finding out that these cells will, of course, give rise to every cell in the body. Well, that's not unexpected. That's what they're designed to do. But in the body where there's a constant draw on cells, you generally have a pool of very primitive cells. Primitive meaning that they're not so differentiated, and they kind of hang out and they give rise to a progenitor. So you have the stem cell and the progenitor cell. So a progenitor cell is an early derivative too, and the spermatogonia are they're a progenitor, a stem slash progenitor type cell. So they're going to give rise to different cell types, and the one that we're interested in is the primary spermatocyte, because that's going to give rise to secondary spermatocytes, spermatid, and then spermatid's going to differentiate into sperm. So all of this happens with spermatogonia, but the spermatogonia is a progenitor cell. So usually what you have to do in a situation with cells that are derived from stem cells is you want to replicate the stem cell, but you also want to give rise to a transit amplifying like cell. This happens in the intestine, it happens in the skin. And the skin is a good example because I study skin and there's a lot of skin. But the epithelia on the skin, it's a stratified epithelia, and it's differentiating as it's moving up, and there are basal cells. And 1 out of 20 of those basal cells is a stem cell. And it replicates itself, but it also gives rise to a transit amplifying cell. The transit amplifying cells are the ones that divide profusely, and they give rise to the whole epithelial layer. And we're sloughing off skin constantly, so there's a draw on cells required. And the lining of the gut is the same way. Constantly need to produce the gut lining over again because it's being sloughed off at a constant rate. Well, sperm are another situation where, where's the cell draw? Well, we were saying a young adult male produces about 200 million sperm every 24 hours. That's enough to populate the whole planet. Every single male produces that. How many eggs does a female produce? Total. We'll talk about that in the next chapter, but in female reproduction, there's about 300,000 eggs that a woman has, but most of those are going to be lost from atresia. There's a process where a lot of the cells, they're not going to be presented as an egg that's ready to fertilize. They're going to lose their viability. They're brought along to a certain level of sophistication, and then the decision is made that we don't need this cell, and so they apoptose basically. But that process is atresia. So, but theoretically, the female carries about 300,000 eggs, and so then most of those are lost by atresia. And you're only interested in presenting in each menstrual cycle in presenting 1 egg or sometimes 2. So that doesn't total up. The number of menstrual cycles doesn't total up to 300,000. So you lose most of those eggs from atresia. But here in the male system is even more prolific and producing many more sperm. So 200 million a day. So how does that happen? Well, the progenitor cells they want to maintain their own population, and they also want to give rise to these other cells that are going to be doing the job, that are going to be dividing rapidly and giving rise to sperm. So, so that's one reason to focus on the spermatogonium, and that's the cell that I'll ask you about. Why does the spermatogonium tend to be near the interstitial cell? It's on the outside. Well, again, it's the duct, right? the things that are ready to be secreted. Actually, even though they've got the heads of the sperm going in this direction, they're actually, once they're released, they have them going in this direction because that's how they're released, but they're going to turn around and start to go out. So that's kind of, they should have drawn some of them going the other way, like turning around, starting to go the other way, because they're going to be going out the duct. So, so there's a constant draw. 200 million of these sperm are going to be produced every 24 hours. So, so there's a lot of replication that occurs here. Some of the replication is mitosis up here, but then there's meiosis down here. So you guys have studied mitosis and meiosis, right? You're all experts on that stuff, right? Yeah. So if the total genetic component of the cell is 2n, for where n is sort of the minimum DNA, then most somatic cells have 2n, and the germ cells have 1n, right? Because you've gone through meiosis, so you have a haploid as opposed to a diploid cell. But when you have the diploid cell and it first divides, when it makes new DNA, it replicates new DNA, so then it's going to be 4n, right, before it actually divides. Yeah. So you're going to go from 4n to 1n for the one copy of a complete genome into each sperm, and it's the haploid genome. And we'll discuss that a little bit more. But this is a main concept, and I want to make sure that you guys are all with me on this, because wherever there's a draw on cells, this will be in the final, and it'll be on the next test a little bit, right? So whenever you have a situation where you need to replenish cells, and especially in a situation like this, where 200 million sperm are being made every day, that you need a lot of replication, so you're going to have this progenitor cell that it doesn't replicate that often, but when it does, it gives rise to a cell that's going to divide a lot, and it also replenishes itself to make sure that there's still a healthy population of the spermatogonia present. And then there on the outside of the seminiferous tubule, they're next to the interstitial cells that are producing testosterone. So the cell that is going to divide is the primary spermatocyte. And that's going to go through meiosis 1 and give you a secondary spermatocyte, and then meiosis 2, and it gives you a spermatid. So I was saying that we don't see, histologically, we don't see the secondary spermatocyte. We looked at those cross sections of the seminiferous tubule, and there was no secondary somatocyte to be seen, spermatocyte. And the reason why there weren't any is because they pretty much divide out into spermatids right away. So there's no lag period there. They don't have to go through prophase. There's no DNA replication. They're just dividing out the DNA further. And so, histologically, you just don't see them. So, but you do see spermatocyte. So instead of labeling it primary spermatocyte, you just label it spermatocyte. Does that make sense? It's actually on the next picture too. This guy here, where this is very similar to that first histological picture that we saw. The cross section of the seminiferous tubules, and they label up the same things. You got the myoid here, which is that single layer of smooth muscle-like cells. Spermatogonia, which tend to hang out on the periphery, and the spermatocytes are here. These are spermatocytes. They don't distinguish the secondary from the primary, because there is typically in a section you don't see any secondary, because we were saying that the cells divide so quickly into spermatids at that point. So then over here, this is where you can see spermatids. The nuclei are very crunched down. So why are these cells so small? Why are the spermatids so small?
Not a lot to them.
Yeah, there's not a lot to them, right? I mean, the spermatogonia, that's the cell that we're depending upon to maintain this fertile, situation where they can keep dividing, but they're going to give rise to a transit amplifying cell, the primary spermatocyte, and the primary spermatocyte this is the one that's going to divide off and give you lots of these spermatids. So the pressure is more on that cell, but then this cell is, the progenitor cell is valuable and a source of cells throughout the cycle of fertility for that male. So these spermatids, they're very small, and you don't want to carry any additional weight. What's the job of a sperm? Sperm has got to swim and find the egg and fertilize, but it doesn't divide, and it's going to do a limited number of things. It will respond to some nutrition. So if you keep sperm in a solution that contains fructose and some electrolytes and some mucus, they can be happy for a couple of days. But they're not really designed to last very long. So they've got their one shot at fame, and so they try to find the egg. That's what they're all interested in. So here's what we're looking at. So the sperm themselves, this is not drawn to scale at all. That's one of the things that they say here. The spermatids are really very small. What is the chromosome number? So it's chromosome 46, 23. Where do the sex chromosomes fit into that? What are the sex chromosomes? Elizabeth, sex chromosomes.
There's 23. Is that what you're saying?
No, there's not 23 sex chromosomes. What is the sexual determinant chromosome? This is, elementary school biology. Christian? Yeah, X and Y.
No.
You can't remember X and Y?
I didn't know you were asking that.
Yeah, so then, when this number, well, just to give you an appreciation of what these numbers mean, so 23, so then in a haploid, there's going to be how many somatic and how many sex chromosomes? Out of the 23. Huh?
What?
Putting you too much on the spot. Catherine, what do you think? Come on, guys. I'm encouraging you to link together all that biology that you've had. So this goes back to your elementary school or maybe junior high school, early high school biology class where they told you, the human cells have 46 chromosomes. And then half that number, the haploid number, 1N versus 2N. So 2N is diploid, so the cells of our body are diploid, and they have 46. And then these sperm have 23. So they have half of that genome. So I'm asking Catherine, out of the 23, how many are somatic and how many are sex chromosomes? Right. Just one is a sex chromosome. The other 22, somatic. Somatic, soma is Greek for body. Somatic, 22 somatic chromosomes and one sex chromosome. So is it X or Y? Right. It's the luck of the draw. It could be X or Y. Okay, I think this is really fascinating here, the development of sperm. there's this acrosomal cap, which is derived from, it's like a lysosome. Inside the spermatid, so we said that we had spermatocytes, but they quickly go through the second meiosis and they're giving you these spermatids. And then the spermatids have this acrosomal cap. The acrosomal cap, it's just like, it's a lysozyme that basically, it's a circular glob, but it ends up being fitted around the head of the sperm. So the nucleus is kind of completely covered by it. It's kind of fascinating. Why? Why do you need the acrosome? Daniel, what's the purpose of the acrosome?
It's to puncture, or not puncture, but enter the exome. And it releases some enzyme that allows the DNA from the sperm to enter into the exome.
Right. So the sperm is searching out the egg, and it wants to combine genetically with the haploid genome that's presented in the egg. And the acrosome is it's folded over the nucleus. It starts out as kind of a lysosomal ball, and it's just, the nucleus is kind of circular, and it's just folded over the top of that, and that is the acrosome, and that structure stays in there in the final form of the sperm, and becomes critical for fertilization because the acrosome it's a trypsin-like enzyme that digests protein. It's able to digest the surface of the egg cell to get in there so that it can inject its DNA. The DNA is in the nucleus. Other things, there's excess cytoplasm, right? You have this larger cell, and cells usually look like eggs. They remind me of like an egg on a frying pan. There's a nucleus, there's some cytoplasm. They look like eggs because eggs are cells, right? They're just one cell, it's one large cell, and they're good with bacon and I don't know. Okay, so in order to develop into a sperm, you need the flagellum. So you're going to start to grow out a flagellum. You have this lysosome kind of developing into an acrosomal cap that has the trypsin-like activity in it. You want to get rid of this extra cytoplasm. You've got extra cytoplasm, but you want to maintain mitochondria. And you want to maintain mitochondria because you want to swim. So you want the flagella to have lots of energy, so then the mitochondria end up lining up along this proximal area of the flagella. And so they're going to be providing the necessary energy for the tail to keep whipping and swimming. And so when you get rid of there's two structural things there. There's the mitochondria lining up along the flagella, and then there's getting rid of this excess cytoplasm. And the excess cytoplasm actually appears as residual bodies. So there's little globs of cytoplasm that will just be eventually shoot up. The other thing about this diagram here is they have in here that there are tight junctions. Tight junctions between the different cells that are in the seminiferous tubule. And so there's a blood-brain barrier, not blood-brain, a blood barrier to the in the testis. So that barrier, all you need to keep fluid from going from one area to another is a tight seal. And so the tight seal is provided by tight junctions here. And so the tight junctions are actually dissolved and then they reform every time a primary spermatocyte is formed. And they bring this up in the book. It's kind of a cool thing. So disassembly of tight junctions is a characteristic of these spermatogonia as they give rise to the primary spermatocyte. It's a lot of work, I think. I guess barriers are made to be broken, but if you permanently damage that, you could have blood getting into the semen, which would not be good. So you want to reform these tight junctions pretty routinely. And there also must be something important about the remaining differentiation of the sperm to not have blood there. Presence of blood indicates a wound for the body. And so I work a lot on this kind of thing, and there are, I think, substances within the blood that signal the body that there's potential there for wounding as well. Unless there's a sinus cavity that's used to receiving lots of blood, then it doesn't perceive that as being a wound. But if blood were to appear where it's not supposed to appear, then the surrounding cells can potentially take that as a signal that there's a wound. Anyway, the tight junction here in these seminiferous tubules are maintaining that barrier between the blood and the rest of the seminiferous tubule. Okay, so when I wrote down important points, the things that I thought were important were that the spermatogonia are progenitor cells, which are stem cell derived, and that they give rise to one spermatogonia, so they regenerate themselves, but they also give rise to a transit amplifying cell, a type of cell that's going to divide rapidly and is the functional cell that needs to be replaced in copious amounts. And so there's a spermatogonia, regenerates itself, but it also produces this primary spermatocyte for subsequent steps, 2 steps of meiosis. And that's just like every other stem cell system in the body. So that's one point that I think is really important. Another central point is that the primary spermatocyte disassembles these tight junctions. So we were just talking about that. It's the blood barrier. As they move from the basal to the adluminal compartment. So we said that this is a lumen. Anytime there's a duct, the central part of the duct, lumen. So the adluminal means it's kind of facing the lumen. So if you move from basal, this basal layer was where the myoid is interfacing between the spermatogonia and these interstitial cells. The third point, DNA replication is kind of critical to this whole process, and the book does not show this very well. So we're actually going to, not on this chapter, but on chapter 3. I don't know if you remember back on chapter 3. So chapter 3, page 70, figure 318. This just shows mitosis versus meiosis. Now this is a histology class, but still to understand reproduction. We end up here. Oh, this is 3. Okay, 318. So this is just the regular mitosis. This is the stem cell replenishing itself, but one of the daughter cells is just another stem cell, but the other daughter cell gives rise to these cells that are needed in large supply. And this is from chapter 3. I guess they're showing you digestive system stuff. But here, this is where you look at mitosis, which is where you've got duplicating chromosomes, and the chromosomes condense, then in metaphase they line up, anaphase, telophase, you get this separation, and you get two identical daughter cells from cell division. But meiosis, you also get replication of the DNA, and then you go through the first phase of meiosis. So meiosis 1 has a prophase and metaphase, where these homologous chromosomes line up and then you separate them. But then you need meiosis 2, where you take these pairs and you pull the pairs apart so that they only have a single copy at that point. So there's only one and just the single genetic copy. But one of the things that's really critical here is that the recombination is so critical. So actually the cells hang out in prophase for like 3 weeks, during which time DNA is exchanged within the chromosomes. Isn't that incredible? So otherwise, you can imagine with the shuffling of single chromosomes that we would still all look pretty much like our parents, or at least kind of things would line up in a very simple way. But because you have this crossover and little parts of chromosomes being able to exchange homologous recombination, so you're giving the cells 3 weeks to reshuffle the DNA, and then you're producing the final chromatids. So that's so I consider that as an essential point to get to, that it's not just a shuffling of the chromosomes as they were. You're allowing extra time for the chromosome, the DNA content in the chromosomes, to reshuffle in much more complex ways, which is why we're kind of a braver experiment. Each generation is a braver experiment in what DNA can do than just reshuffling in a simple way what the parents are. The fourth important point was that the Sertoli cells are so critical as a supportive cell. And so we've looked at that before, like with neurons, for example, and glial cells, where you had so many more glial cells. In male reproduction, the Sertoli cell is a critical support cell, and you wouldn't get maturation of spermatids without that. The fifth point was, I think the acrosomal cap is really interesting. The fact that it's a lysosome derived organelle that is involved with breaking through into the egg, and it has a trypsin-like enzyme that allows it to digest a protective layer on the egg and therefore fertilize the egg. Sixth thing is that I think the flagella are really interesting, that the mitochondria circling the mid-piece, that area underneath the acrosomal cap, and that you get rid of all the other cytoplasm. So those are all things that I think are kind of key points about this process. Spermatogenesis. And then spermiogenesis is where you're going from the spermatid to making sperm, and that was with the thing with the acrosomal cap and getting rid of the extra cytoplasm and arranging. So it's actually the morphology of the sperm itself comes about during the spermiogenesis, whereas spermatogenesis is the derivation of spermatids from the spermatogonium. there's more that needs to be looked at here, physiology-wise. Anyway, so go back to chapter 21. What kind of picture is this? EM, right? Catherine Tran, what kind of EM? Yeah, it's an electron microscope, but that's EM. But what kind? We had scanning and another one. What was the other one? TEM. What does the T stand for? Transmission. Okay, so this is a transmission electron micrograph. So the transmission was, remember, that was the one that was done with sections, right? So the scanning EM is where you bounce electrons off of a surface, and usually the magnification is lower on that, but you can really see in fine detail surface characteristics. Transmission electron microscopy is done with a section, and you usually have very high power, and you're looking at these organelle-like structures inside of the cell. You can really pick up the morphology of tiny organelles. What is the, how big is a molecule compared to this? If you look at the higher end of the magnification with a transmission electron microscope versus a molecule, are we in the same ballpark, or are we still a billion times away? Catherine, Castro, what do you think? Would you see a molecule on a picture like this? Or would you need to be 10x, 100x, 1,000, million, a billion more power to see a molecule? What's that? You wouldn't see it. So it's more than a billion, you'd need more than a billion-fold further magnification to see a molecule from a transmission electron micrograph. No. Actually, you can see molecules at this level. So, and I think that that's an important point, because the, the physiology of the cell, the organelles, what's going on inside the cell, the cells are dealing with molecules, and they have to be the right size to deal with a molecule. So when you're looking at these little structures, you're looking at structures that are made up of molecules, and they trade molecules. And remember when we were talking about the filtration system, pancreas and filtration, and so we were saying that the gaps that cells can make are just the right size, that they can exclude certain molecules. They can exclude larger proteins versus smaller ones. Sometimes they can exclude even ions. And there are ion transport, pores and specific things to deal with ions, because they're very small. But I mean, the cells, even physically, they can form gaps that are so small that they filter certain size molecules. So these organelles in a cell, they're really at the level of molecules. We're right in that ballpark. So anyway, what is the purpose of this picture? The head of a late spermatid during spermiogenesis. And it's completely enclosed by Sertoli cell cytoplasm. So here's the spermatid, and then I guess that's its little flagella there. But all the rest of this stuff is all the cell stuff from the Sertoli cell. So you can see how intimately the spermatids, although they're not deriving from the Sertoli cell, they're not hatched in the Sertoli cell, they're hatched from the spermatogonium, but they have to travel. Their cytoplasm is so intimately associated with it, they gain nutrition, and they're allowed to develop on the surface of these Sertoli cells, and they're really dependent upon them. They wanted to point out that there's some special transport vesicles here that are bundles of microtubules and actin filaments called manchette transport. Here, just to show you that, yeah, there's a lot of complexity to this. there's always other things that you can look at. So that's not the whole story, though. So we still have to make our way out through the male reproductive organ thing. So we spent a lot of time looking at the testes. So we started out with that, and we've been here the whole time, right? We spent, an hour and a half or something just thinking about the testes. But, remember that we've got this whole morphology to deal with. And, I think biologically the action is really here. And so we've covered that. But we're going to take a look at some of the, what some of these other, the morphology of the cells and some of these other parts of the male reproductive organs look like. So the first thing would be, we need to make our way out of the seminiferous tubules. So the seminiferous tubules is the thing that we've spent a lot of time on. And we're saying that they have these myoid, which is like smooth muscle, and there's a gentle contraction, squeezing out some developed sperm. And the sperm are going to initially reside in the epididymis. And they're going to be making their way up through the ductus deferens when needed. And they're going to be mixed with fluid made in the seminal vesicle. And then that's going to be, it has to make its way out, reproductive organ. So what does the epididymis look like? So that's what they show here. So again, coiled, long duct. Sperm can undergo further maturation, short-term storage. There's going to be a lot of blood vessels. So V here, blood vessels. Again, any kind of duct looks like lots of epithelia surrounding the duct. That's over in the next picture. They're showing you the columnar epithelia of the epididymal duct surrounded by a thin circular layer of smooth muscle. So although the seminiferous tubule doesn't have much smooth muscle, once you get into the epididymal duct, you're going to need a little bit more contractile ability. And in the center here, that's going to be sperm. Sperm. So this is the epididymis. And why is there stuff in the middle? Well, that's sperm in the middle. Smooth muscles, thicker. Okay, so the next thing we're going to look at the vas deferens. And that looks like this. So remember, coming out from the testes, there was that tube that's making its way up, and then it's going to connect into the plumbing. With the urethra and has to go through the male sexual organ. So that big tube, it's we're not dealing with a cell level thing anymore. We're dealing with a much larger tube. And this is what a cross section of a larger tube looks like. So, this is a good one to look at because, this is something that I would want you guys to be able to identify too in a picture. So if I show you a picture like this, Well, what you can say firstly is that this is fairly low power, right? I don't see individual cells so much, but it looks kind of grainy, so those are the individual cells actually. And the thing in the middle is what, Stephanie? The lumen. Right, so there's the opening, vas deferens, and so, but you have a much thicker tubule, so there's a lot here, a lot of other cell material, unbelievable amounts of material. Well, what is all that stuff? Well, M is mucosa, so just a little bit here. So you want to have a mucal secretion there because the sperm haven't mixed with anything. They're coming out of the testes. They were collected in the epididymis. They don't have a fluid. They don't have the seminal fluid yet. So you want to have some. Everything needs to be coated with mucus. So you have a mucal layer of the mucosa, and then you have a thick muscularis layer, and it's actually several different layers. So there's an inner and outer layer of longitudinal muscle. So the L is longitudinal, and the C is circular, and then again another longitudinal. So you see these three thick muscular layers, and they're all smooth muscle. And then the mucosa is the one that's near the lumen, and that's the vas deferens. That is that big tube coming out of the testes that's going to be transporting all of that, all the sperm. So then, looking at this. So this is a frontal picture of that plumbing that we were looking at. The first diagram we had looked at was a side section. Here we're looking straight on. Of course, this is not in proportion exactly, but things are kind of flattened out just to show you. So we spent a lot of time with testes and epididymis. We looked at the ductus deferens, and that had a lot of different muscular layers as well as that mucosal layer. And that brings us all the way up to here. And so this is where we're going to get the seminal vesicle here, and this is going to add fluid to the sperm. And then it needs to be pumped out. You have a prostate gland here. And the urethra then goes through the penis, and penis has a couple of different layers. The corpus cavernosum is this outer layer, and then the corpus spongiosum is an inner layer. And then there's the urethra. It's a spongy urethra right down the center, and the head of the penis is the glands. So, seminal vesicle, kind of an important item, right? Because the real biology is with the sperm and storing the sperm and getting it to go up the ductus deferens. But this seminal vesicle here is going to add semen, all of that fluid it's going to support the sperm, keep it alive, and you want to, it's going to increase the volume of the ejaculate, right? right? So that what does this look like? It's a gland and it's producing lots of fluid. So what would you think that would look like in a cross section? Well, it looks like that. So they're an exocrine gland that secretes most of the seminal fluid, sperm nutrients. What's this thing L in the center? Christian? Yeah, it's the lumen, right? Yeah, it looks like a gland. Looks like exocrine gland. So this is a very low power picture of what the seminal vesicle looks like. If we look at higher power, that's the next picture over. It's got some smooth muscle surrounding it because it has to squirt, right? It's gonna, it's a gland, and you need this fluid to be combining with sperm at the right time. So you're going to need some smooth muscle to help propel that fluid out. There's type of cells that are involved here. These are smooth muscle cells, a lamina propria, LP. Okay, what about the prostate? So, the prostate. So we just looked at the seminal vesicle here, and now we're going to look at this prostate gland. And they show you, this is what a section of the prostate looks like. You've all heard of the prostate gland. Males tend to have problems with their prostate glands. As they get older, they can get prostate cancer. PSA, prostate-specific antigen, is produced in the prostate gland, and PSA has an important functional role, actually, but it's used as an indicator. If there's too much PSA, the PSA levels are too high, then the prostate is making too much of it, therefore indicative that there's something wrong with the prostate gland. Let's see if we can, here's the prostate gland. So this is, you see that the seminal vesicle here is adding fluid. The vas deferens is where we have the sperm coming in. As they're connecting it into the prostate, those are labeled ejaculatory ducts. And the prostate is going to have an outer layer, a peripheral zone, a more central layer, central zone, and a transitional zone surrounding the prostatic urethra. And of course, everything's going to be coming out of the urethra. So what does that look like? It looks like this. It's basically fibromuscular stroma. So it's very dense, and it's what you'd expect. It looked like a big mass of muscle, right? It has to propel material out fairly vigorously. So it's not surprising. There's also large number of small tubulo-alveolar glands, labeled G here. Higher magnification picture of the same thing is shown over here. And that's just showing one of these tubular alveolar glands surrounded by epithelia. The epithelia here and LP lamina propria. Okay, that's enough detail on that. So, the structure of the penis. It it's going to have, it's an organ that has a pretty simple function. It's going to need to be innervated, so you're going to have veins, arteries, and nerve. So we're used to seeing this kind of accompanying arrangement to all the internal organs. And basically a penis has to get erect, so you're going to need a corpus cavernosum. Here there are two of those, one on each side. And then there's a corpus spongiosum here on the bottom, and that's, the spongiosum is wrapped around the spongy urethra, which is where the plumbing is, either urinary or sperm coming out of there. So so that's a cross section. So then how does that work? Here's a section of an actual section. The other was a cartoon. This is an actual section. And in order to get erect, the penis needs to fill with blood. And so the cavernosus space in all three of those masses, the two on the sides and the corpus spongiosum, they fill with blood. And that's on orders from the central nervous system and autonomic nerves in the vascular wall. And as they fill with blood, it also constricts arteries and trabecular muscle and increases blood flow into the spaces, lowering the pressure there, allowing the veins to drain most blood. So this filling with blood causes the erection, and then the releasing of that blood would allow that to pass. Here's the final picture showing the corpus spongiosum here. Penile urethra. Then this is a higher magnification. Erectile tissue is shown. Small portion of the tunica albuginea and fibrous elastic connective tissue. So when these spaces are filling with blood, it causes compression of venous drainage. So therefore, that's why the blood is trapped in there. Does that make sense? So not only does it fill with blood, it also, by filling with blood, it stops the veins from allowing the blood to escape, and so it stays in that condition until otherwise instructed. Okay, so that's it. Any questions on this stuff? So next week we're going to talk about female reproduction. For today, I actually wanted to look at male sperm. So I have to go. I was going to go and check upstairs to see what the department chairman thought of that idea. We'll see if that goes through or not. If not, I'm interested in your sea urchin possibility. I don't know who owns those sea urchins. You know who those are? They must, maybe they're
It might be that same class, yeah.
It's which class?
I think it might be, I think there's two classes at the same class.
Yeah, we have developmental biology, we use them, but I think we're done.
Okay, so did you do that lab?
Yeah, we did it.
Oh, you did it?
That's why I meant.
Okay, and it was really cool, right?
Yeah.
Yeah. So, yeah, so that's interesting. Okay, well, yeah, so maybe, have you guys already had that lab then? You all have. You haven't. So that's why I was thinking, looking at male sperm as from a human is kind of different. Something we should do in this class, because in the developmental biology you've looked at that before. But anyway, I'm going to go upstairs for a minute. So you have a break for, let's say we reconvene by 3:40, and I'll tell you what the department chairman says on that. Any thoughts on that? I just think because this is it's human biology and it's human histology, that it's interesting. And since every male, especially young males, are making 200 million every 24 hours, that it's not really difficult to get, and it's not infectious either, unless ultimately infectious. But it's not like blood, so it's not going to pass hepatitis or anything. All right, let's reconvene at 3:40.